The liver is an accessory organ for digestion. It manufactures bile and secretes it. That bile is going to drain out through the ducts, and it will be poured out into the second part of the duodenum for the purposes of digestion of fats. Of course, bile has other functions, but for digestion of fats is the principal function of bile for digestion. So it's manufactured by the liver, drained out through the bile ducts, all known as the hepatic ducts. We shall look at them. Stored in the gallbladder, and when there is fat in the duodenum, it is released for the purpose of digesting fats. Metabolism of carbohydrates is the other function of the liver. You remember the liver can convert glucose into glycogen and vice versa to maintain the liver cells. The liver cells with the intermediate of insulin convert glucose into glycogen In fact, they convert glucose so that they maintain the blood glucose level to a homeostatic balance. And we shall look at that in detail. The liver manufactures proteins which are very, very vital in the body. They are very, very vital in the body. Vital proteins which are very important for the normal functioning of the body. Some of these proteins are clotting factors, and some are carrier protein. Produces bile and secretes it. When this bile leaves the liver through hepatic ducts, it is stored in the gallbladder, and the gallbladder will concentrate it. When you have fatty food in the duodenum, and this bile is released from the gallbladder, and it will go and enter the duodenum to digest fats. So the purpose of the liver and digestion in this respect is to produce and secrete bile, which will be used in the digestion of fats. Metabolism of carbohydrates, for example, the liver converts glucose to glycogen, and that process is stimulated by insulin to lower blood glucose if it has gone beyond the normal level. And then when blood glucose drops through the intermediate of glucagon, the liver can convert that glycogen back into glucose. And those processes are to maintain a homeostatic balance of glucose. So both glucose and glycogen are carbohydrates, and their metabolism is mediated by the liver to maintain blood glucose. Metabolism of fats, build up of fats. The liver can manufacture fats, and these fats are storage of energy. They store energy and then they can be broken down to release that energy. Metabolism of proteins, the liver manufactures vital proteins. Some proteins are clotting factors, some are plasma proteins, and we talked about the importance of plasma proteins. The liver manufactures those proteins, which are very vital for the normal functioning of the body. So it participates in the metabolism of carbohydrates, fats, and proteins. Then the other function of the liver is to filter blood by removing bacteria and toxic substances. These functions make the liver a vital organ without which you cannot survive. And you're going to go and break them down and read all these functions in details, seeing how the liver manufactures bile, the metabolism of carbohydrates by the liver, protein by the liver, and fats by the liver, then the process of the filtration of blood, which is also carried out by the liver. These are vital functions of the body. The liver is below the thoracic diaphragm, and the thoracic diaphragm is molded by the lobes of the liver. That is why you have by the lobes of the liver. That's why you have the domes of the diaphragm. The domes of the diaphragm are convex on the thoracic side, and they are concave on the abdominal side. They are convex on the thoracic side and they are concave on the abdominal side. What is causing that? Below the thoracic diaphragm, we have the lobes of the liver. And those lobes of the liver, superiorly, they are convex. That's why they mold the thoracic diaphragm into those domes. So the liver is located below the thoracic diaphragm. Its upper surface is convex. So if its upper surface is convex, it is going to push the domes of the diaphragm up into the thorax. So that upper surface which molds the domes of the diaphragm is the superior one in its contact with the diaphragm, and the inferior surface of the liver is in contact with abdominal organs or structures. So the inferior surface of the liver, which is in contact with abdominal structures, is known as the visceral surface because it is in contact with the viscera, which are the structures of the abdomen. And the upper surface, which is convex, is in contact with the diaphragm. Now, this is where attention is required when we are talking about the lobes of the liver. So when we talk about the lobes of the liver, we must be very attentive because they are classified in different ways. So when we are talking about the lobes of the liver, we have what we call anatomical lobes. And they are also what we call physiological or functional lobes. So you can classify the lobes of the liver into anatomical lobes, and you can also classify the liver into functional or physiological lobes. So our task is to know what the anatomical lobes are and what the functional lobes are. Because those two are different, functional lobes and anatomical lobes. That's why we need to pay attention when we are talking about these lobes of the liver. So I am projecting an illustration, and I hope all of you are seeing it. And we are seeing on the upper left our liver below the thoracic diaphragm. And we are seeing that the upper surface is convex, and that is what molds the liver into its domes. The inferior surface is the one which is in contact with the abdominal structures. That's why they call it the visceral surface. So when we get this liver out of the body and we magnify it here, we are seeing the upper surface, which is convex, and the inferior surface, which is below there, which is known as the visceral surface. So this is the anterior surface of the liver, and the inferior surface is below there. So when you turn the liver upside down to visualize this inferior surface, this is how it looks like. This is the inferior surface. And so we have brought out this liver to magnify it so that we can see it in detail. The surface below there is the one which we are demonstrating here. We have turned the liver upside down to show you the inferior or visceral surface. This is the anterior surface, and above there, that is the superior surface, which you have magnified here. This is the superior surface. So this is showing us the inferior surface of the liver, which is below there. This is showing us the superior surface of the liver, which is above there. And then this is the anterior surface of the liver. Now, let's proceed. This is peritoneum. This peritoneum is known as the falciform ligament. Falciform ligament. How does the falciform ligament form? Peritoneum peels off from the anterior abdominal wall. Peritoneum peels off from the anterior abdominal wall. It goes deep into the abdomen and attaches on the anterior surface of the liver. So this falciform ligament is peritoneum, which is peeling off from the anterior abdominal wall and going deep into the abdomen, where it reflects itself onto the liver. So it is made up of two membranes, one on the left, one on the right. These membranes peel off from the anterior abdominal wall, they go deep into the abdomen, and then they reflect themselves onto the anterior surface of the liver. So those two membranes of the peritoneum, which are peeling off from the anterior abdominal wall, are the ones that we call the falciform ligament. Within the falciform ligament, we have the round ligament of the liver, or ligamentum teres hepatis, running through the deep aspects of the falciform ligaments, moving towards the liver. So within this falciform ligament, you have the ligamentum teres hepatis, or the round ligament of the liver. This is coming from the umbilicus, and it is connecting to the liver. So what is this round ligament which is running through the falciform ligament? Who remembers the function of this falciform ligament? Have I ever talked about falciform ligament?
It is
The round ligament, or
the
the
liver to
ligamentum
the
teres?
anterior wall. It is to attach the liver to the anterior abdominal wall.
What was it before birth? The round ligament. You either call it round ligament or ligamentum teres. What was it before birth?
Ductus venosus.
No. Ligamentum teres moves from the umbilicus, passes through the falciform ligament, and goes to the liver.
It was the.
And I talked about it.
Was the.
Was the umbilical vein. The left umbilical vein.
So this ligamentum teres, before birth, it was the umbilical vein, which was picking oxygenated blood from the placenta, passing through the umbilical cord, entering the abdomen, and taking it to the liver, where it connected with the ductus venosus. So at birth, the umbilical vein closes to form the ligamentum teres. So in adult life, we shall see this ligamentum teres running through the falciform ligament towards its way onto the liver. So this falciform ligament is peritoneum peeling off from the anterior wall of the abdomen, getting deep into the abdomen and reflecting itself onto the liver. Within it, we shall see the ligamentum teres also running, which is our obliterated umbilical vein. In fetal life, it was the umbilical vein. At birth, it closed to form the ligamentum teres. Now we see it running through the falciform ligament. Now, every point is very important, because someone may ask why I'm spending a lot of time talking about the falciform ligament. This ligamentum teres is visible on ultrasound. But here we are seeing its position running through the falciform ligament towards the liver, and it is the obliterated umbilical vein. And we have it in adults. So it is between the umbilicus and the liver, but it's running through the deeper aspects of the falciform ligament. Now, this point where the falciform ligament reflects itself onto the liver, you can see this point. This is where the falciform ligament reflects itself onto the liver. On the right, this is the right anatomical lobe, and on the left, this is the left anatomical lobes. So when we divide the liver into right anatomical lobe and left anatomical lobe, remember the word anatomical is very important. The right anatomical lobe is on the right side of the falciform ligament, and the left anatomical lobe is on the left side of the falciform ligament. Those are the anatomical lobes. That's point number one, which you should never forget. Now let us look at the inferior or visceral surface of the liver. This is the inferior surface of the liver. And we can see the boundary between the right, this is the right anatomical lobe, and then this is the left anatomical lobe. Because the left anatomical lobe is on the left side of the falciform ligament, and the right anatomical lobe is on the right side of the falciform ligament. That's the point I have made. Now, when you look at the inferior surface of the right anatomical lobe, this is the right anatomical lobe. The inferior surface of the right anatomical lobe has other features. We are having this point there. This point here, you can see it is surrounded by peritoneum, is what we call the porta hepatis. Now we are on the inferior surface of the liver, focusing on what we see below the right anatomical lobe. And we are seeing the porta hepatis. This porta hepatis is the point of entrance of the portal vein, which is colored blue, of the hepatic artery, which is colored red, and the exit of the bile ducts, which are colored green. So blood enters the liver on the inferior surface at this point known as the porta hepatis. Portal vein is entering, hepatic artery is entering, and bile ducts are leaving. This is the porta hepatis, the point of entrance of the blood vessels and exit of the bile ducts. The porta hepatis can also be known as the hilum of the liver. So you can say the hilum of the liver or the porta hepatis. Now, this peritoneum, which is surrounding the structures of the porta hepatis, is the hepatoduodenal ligament. Now, let me show you something so that you know that whenever we are doing anatomy, these things are connected. You remember I told you that peritoneum peels off from the undersurface of the liver and it attaches to the lesser curvature of the stomach. What did we call this peritoneum? Who can remember the name of this peritoneum? Peeling off from the liver and attaching to the lesser curvature of the stomach. What name did we give this peritoneum? I talked about it when I was talking about the stomach.
It is called the lesser omentum.
It is called the lesser omentum, correct. And I said that the lesser omentum has two parts. Part of it is the one which peels off from the liver and attaches to the greater curvature of the stomach. And part of it peels off from the liver and attaches to the first part of the duodenum. So what did we call this part of the lesser omentum which attaches to the lesser curvature of the stomach? Does anyone remember?
Can I say that?
Hepatogastric ligament.
Hepatogastric ligament. That is part of the lesser omentum between the liver and the lesser curvature of the stomach. And this one, which is between the liver and this part, which is between the liver and the first part of the duodenum, what do we call it?
Hepatoduodenal ligament.
Hepatoduodenal ligament. So here we are seeing that this is our falciform ligament containing the ligamentum teres. So this is our right anatomical lobe and this is our left anatomical lobe. So below the right anatomical lobe, we have the hepatoduodenal ligament. Below the right anatomical lobe, we have the hepatoduodenal ligament, which contains the red hepatic artery, the green bile duct, and the blue portal vein, this one there. And this point where the hepatic artery, the portal vein, enter the liver and the bile ducts leave it, this is what we call the porta hepatis. That's where structures enter or leave the liver. And those structures at the porta hepatis are surrounded by the hepatoduodenal ligament on the inferior or visceral surface of the liver. That is exactly what we are seeing here now. So we can see this hepatoduodenal ligament on the inferior surface of the liver, surrounding the portal vein, which is colored blue, hepatic artery, which is colored red, and the bile ducts, which are colored green. And this peritoneum surrounding them is the hepatoduodenal ligament. So when we look at the inferior surface of the liver or the visceral surface of the liver, we are seeing our falciform ligament, indicating that this is the right anatomical lobe and this is the left anatomical lobe. Then we are seeing below the right anatomical lobe the porta hepatis, or the hilum of the liver, where blood vessels enter and the bile ducts live. And these structures of the porta hepatis are surrounded by the hepatoduodenal ligament. What else are we seeing below the right anatomical lobe apart from the porta hepatis? We are seeing the gallbladder. The gallbladder is on the inferior surface of the right anatomical lobe. In fact, the peritoneum, which is lining the liver, seals the gallbladder on the undersurface of the liver. That is why when you move the liver, it moves with the gallbladder, because peritoneum of the liver has sealed it on the undersurface of the right anatomical lobe. So we are seeing the gallbladder on the inferior surface of the right anatomical lobe. Remember the word anatomical lobe. What else are we seeing? We are seeing a subdivision there. The subdivision is just there. See, between the gallbladder and the falciform ligament, the subdivision there. So this is the subdivision. This subdivision on the inferior surface of the right anatomical lobe is known as the quadrate lobe. So the quadrate lobe is a subdivision of the right anatomical lobe, and it is visualized on the inferior surface of the right anatomical lobe of the liver. On the right, it is bounded by the gallbladder. On the left, it is bounded by the falciform ligament and the ligamentum teres. And posteriorly, it is bounded by the porta hepatis. I repeat, I said that on the inferior surface of the right anatomical lobe, we have a subdivision known as the quadrate lobe, because it is four-sided. And this quadrate lobe is a subdivision of the right anatomical lobe. It is bounded on the right by the gallbladder, on the left by the falciform ligament, which is containing the ligamentum teres, and posteriorly by the porta hepatis, which is surrounded by the hepatoduodenal ligament. That is the quadrate lobe. Then posterior to the porta hepatis, we have another subdivision here. This subdivision is known as the caudate lobe. So the caudate lobe is another subdivision of the right anatomical lobe, which is visualized on the inferior surface of the liver. So what else are we seeing apart from the caudate lobe? We are seeing that on the right of the caudate lobe, you have the inferior vena cava. You see that inferior vena cava passes posterior to the liver, which we have seen here. So on the right of the caudate lobe, we have the inferior vena cava. And on the left of the caudate lobe, we have the ligamentum venosum. That is the ligamentum venosum. On the left of the caudate lobe, we have the ligamentum venosum. On the right of the caudate lobe, you have the inferior vena cava, and anterior to it, you have the porta hepatis. Who can tell me what the ligamentum venosum was in fetal life? In fetal life, who can tell me what the ligamentum venosum was?
It was the ductus venosus.
It was the ductus venosus. Blood came through the umbilical vein, then entered the ductus venosus. At birth, the umbilical vein becomes the ligamentum teres, and the ductus venosus becomes the ligamentum venosum. So, on the inferior surface of the liver. By the way, the inferior surface of the liver is not flat; it is oblique. It is oblique. That's why you are able to even to see the inferior vena cava, which is running posterior. So, on the inferior surface of the right anatomical lobe, we have managed to see the gallbladder, the porta hepatis surrounded by the hepatoduodenal ligament and its content structures, the ligamentum teres in the falciform ligament, the caudate lobe, and the caudate lobe. Boundaries of the caudate lobe on the right, inferior vena cava, on the left, ligamentum venosum, and anteriorly, porta hepatis. And boundaries of the caudate lobe, on the right, we have the gallbladder, on the left, we have the falciform ligament, which are containing the round ligament of the liver, and then posterior you have the porta hepatis. And then you can see that this porta hepatis is the anterior to it you have the caudate lobe, posterior to it you have the caudate lobe. So there is a lot to see on the inferior surface of the right anatomical lobe of the liver. But you can still see other depressions. These impressions are brought about by the attachment of the undersurface of the liver to the viscera or to the abdominal structures. They are the ones which form these depressions that you see on the inferior surface of the liver. So this is very important image because we are seeing structures on the inferior surface of the right anatomical lobe. The inferior surface of the left anatomical lobe is not so eventful, but still there are structures which cause these impressions. Now let us look at this upper surface. Now, when the falciform ligament attaches to the anterior surface of the liver, forming the boundary between the right anatomical lobe and the left anatomical lobe, on the superior aspect, this falciform ligament splits into two. The membrane on the left reflects towards the left, and the membrane on the right, membrane of peritoneum on the right, reflects onto the right. So it's splitting into two towards the superior surface. So these two peritoneal membranes, which are as a result of the splitting of the falciform ligament, are known as coronary ligaments. So here we are seeing the falciform ligament splitting into the left coronary ligament and then the right coronary ligament. One of the importances of these two coronary ligaments is that they attach the liver to the diaphragm, which is above there. These coronary ligaments attach the liver to the thoracic diaphragm. And let's see, there is an illustration that I want us to see and appreciate. So you can see the falciform ligament is dividing into the right coronary and the left coronary. You can see they are now attaching the liver onto the undersurface of the diaphragm. The right coronary extends up to the extreme right, and the left coronary extends up to the extreme left. We might not be seeing it here. So this is what I really mean by these coronary ligaments. The first form towards the superior aspect of the liver splits into two, the right coronary, which moves up to the extreme right, and the left coronary, which moves up to the extreme left. And they attach the liver onto the undersurface of the thoracic diaphragm. That is why the liver, much as it is the largest organ, it is up there, because the coronary ligaments spill off from the liver and attach it to the diaphragm, hence supporting it, so that it doesn't drop down. But we shall see other supports apart from the ligaments. So these are what we call the left coronary ligament and right coronary ligament, which are as a result of the bifurcation of the first form ligament towards the superior surface of the liver. And these coronary ligaments, they move up to the extreme left and extreme right as they attach the liver onto the undersurface of the thoracic diaphragm. So the same applies here. This is our false form ligament there. And this false form ligament, you can see it is there. It's dividing into the left coronary, which is here, and then the right coronary, which is there. So our eye is up here now. We are seeing these two ligaments. Now, what happens when the left coronary reaches the extreme left and the right ligament reaches the extreme right? So this point is the point we are seeing there, and this point is the point we are seeing here. So when these two ligaments reach the extreme left and the extreme right, they bend back. They form a V. They bend back like that. They bend like that, which we are not seeing here, but the bend is there and the bend there. So they curve back on the posterior aspect and they move like that. So these curves are the ones we call triangular ligaments. So at the extreme end of the coronary ligaments, there's a bending which forms the right triangular ligament, and on the extreme left, there is another bending which forms the left triangular ligaments. So here we have the left triangular ligament, which is there, and here we have the right triangular ligament, which is there. The coronary ligament on the right is this one, and the coronary ligament on the right is that one. Both the triangular and coronary ligaments on the right and on the left participate in the attachment of the liver onto the undersurface of the thoracic diaphragm, whereas the false form ligament attaches the liver onto the anterior abdominal wall. So the liver is attached onto the thoracic diaphragm by the right and left coronary ligaments and then right and left triangular ligaments. And it is in turn attached onto the anterior abdominal wall by the false form ligament and the round ligament of the liver. Hence, the liver is fixed in position by its attachments onto the thoracic diaphragm and onto the anterior abdominal wall. So you can see this slide had volumes to speak. And we shall go back and make an assessment of those features that I have talked about in this illustration. Talking about the anterior surface, the inferior surface, the superior surface, the right anatomical lobe, the left anatomical lobe, the first form ligament, and the features on the undersurface of the liver, gallbladder, portal hepatis, first form ligament, caudate lobe, ligamentum venosum, inferior vena cava, and the rest. How the liver is attached onto the thoracic diaphragm and how the liver is attached onto the anterior abdominal wall to fix it in position. Don't forget that. These are the same. You can see the first form ligament and the ligamentum teres, and then These are the triangular ligaments. So before I proceed, from what I have said, these colors, I'm going to talk about them. The liver is further divided into segments. So here we have used colors to make it easy. These are what we call segments, and I'm going to talk about the segments of the liver. At the moment, I've talked about anatomical lobes. I've not yet talked about physiological lobes, and I've not yet talked about segments. So after talking about the features of this slide, anatomical lobes and their other features, now the next point, I am going to talk about functional lobes. Sometimes the functional lobes are known as physiological lobes. How are functional lobes different from physiological lobes? And as I said, you have to pay attention. How are functional lobes different from anatomical lobes? That's the question. So I was saying that the next step we are going to look at are the functional lobes of the liver. The functional lobes are also known as physiological lobes. The functional lobes are also known as physiological lobes. Remember, the anatomical lobes are demarcated depending on which side of the falciform ligament it is located. So the basis of the anatomical lobes is dependent on which side of the falciform ligament the lobe is located. And for the anatomical lobes, we said on the right of the falciform ligament, right anatomical lobe. On the left of the falciform ligament, left anatomical lobe. Now, what about the functional lobes? On which basis are they demarcated? These are based on the blood supply. When these blood vessels reach the portal hepatis, for example, when you look at the portal vein and the hepatic artery, when they reach the hilum of the liver, they divide into right and left. Let us first, I'm going to show you another illustration because we want to know the basis on which the lobes are demarcated so that we appreciate it. So when you look at these blood vessels, this is the portal vein, the one colored blue, and this is the hepatic artery, the one colored red. When they reach the portal hepatis, they divide into left and right. The artery divides into left and right. Even the vein divides into left and right. So these blood vessels, when they reach the portal hepatis, they divide into right and left. Now, part of the liver which is supplied by blood in the left vessels, that part is known as the left functional lobe. And part of the liver which receives blood from the right branches, that is known as the right functional lobe. So the functional lobes are dependent on the blood supply. Part of the liver which receives blood from the right branches, that is the right functional lobe. And part of the liver which receives blood from the left branches, that is the left functional lobe. So for example, the left branch of the hepatic artery is taking blood to the left functional lobe of the liver, and the right branch of the hepatic artery is taking blood to the right functional lobe of the liver. So where are our functional lobes as opposed to the anatomical lobes? When it comes to the functional lobes, and this is very important, the caudate lobe and the caudate lobe are taken to be part of the left functional lobe. So the whole of this is the left functional lobe. And then the rest of the liver on the right of the gallbladder is what we call the right functional lobe. Now you can even see these arrows. The whole of this is the left functional lobe up to this point, and the remainder is the right functional lobe. So we say that this is the left functional lobe because this is the part of the liver which receives blood from the left hepatic vein and left portal vein and left hepatic artery. They supply this part of the liver. So this is the left functional lobe. And then this other part on the right of the gallbladder is the one which receives blood from the right hepatic artery and then the right portal vein. That's why this is the one we are calling now the right functional lobe. Now you can see that the right functional lobe is smaller than the left functional lobe. But on the other hand, the right anatomical lobe is bigger than the left anatomical lobe. So when I'm talking about the functional lobes, this part of the right anatomical lobe, which contains the caudate lobe and the caudate lobe, is added onto the left anatomical lobe, and together they are going to form the left functional lobe because these parts receive blood from the left portal vein and the left hepatic artery, making the functional lobes left one bigger than the right one. So the boundary of the functional lobes is here between the gallbladder and the vena cava. It is imaginary. So when you make an imaginary line from this point of the gallbladder and take it up to that point where you see the inferior vena cava, this imaginary line is where you have the boundary between the left functional lobe and the right functional lobe. But the falciform ligament is the boundary between the right anatomical lobe and the left anatomical lobe. It means that the right anatomical lobe is bigger than the left anatomical lobe, but the right functional lobe is smaller than the left functional lobe. And we say that functional lobe are demarcated on the basis of the blood supply. The parts of the liver which receive blood from the left portal vein and the left hepatic artery are known as the left functional lobe. And the parts of the liver which receive blood from the right portal vein and the right hepatic artery are the ones which we consider to be the right functional lobe. And the boundary between the left functional lobe and the right functional lobe is an imaginary line between the gallbladder and the inferior vena cava. And that imaginary line, we call it the main lobar fissure. The imaginary line between the gallbladder fossa and the inferior vena cava, we call it the main lobar fissure. Main lobar fissure, and it's the boundary between the left functional lobe and then the right functional lobe. So those are the functional lobes. Here we have indicated the functional lobes as opposed to the anatomical lobes. And here you're seeing that the anatomical lobe on the right is bigger than the anatomical lobe on the left. And then here we're seeing the functional lobe on the right is smaller than the functional lobe on the left because of what I have explained to you. So at the moment, I have finished talking about the functional lobes and the anatomical lobes. I have also talked about the porta hepatis, or the hilum of the liver, and that part of the lesser omentum known as the hepatoduodenal ligament, which surrounds it, and the structures which are located there. On the inferior surface of the liver, that's where you have the porta hepatis. That's the point of entrance of blood vessels and exit of bile ducts. So I talked about the lesser omentum, and here we have been focusing on the hepatoduodenal ligament because it's the one which surrounds the structure of the hilum of the liver. Posterior to the hepatoduodenal ligament, there is an opening. You can see that arrow entering a space posterior to the stomach and the lesser omentum. That space posterior to the stomach and the lesser omentum, I talked about it last time. It is the lesser sac, but you enter into it through an opening known as the epiploic foramen. So that epiploic foramen is posterior to the hepatoduodenal ligament, and it is the entrance into the lesser sac, which is the space posterior to the lesser omentum and the stomach. So here what we have done in this illustration, we have peeled away part of the hepatoduodenal ligament to show you that inside, that's why you have the hepatic artery, the bile duct, and the portal vein. So what has been done here, they have peeled away part of this hepatoduodenal ligament to show you the contents which enter or leave the hilum of the liver. And if you observe, the hepatic artery is on the left, the bile duct is on the right, and the portal vein is posterior to both of them. This arrangement is also important. We just don't say that structures at the hilum of the liver have the hepatic artery, the portal vein, the bile duct. Which arrangement? The hepatic artery is on the left, the bile duct is on the right, and the portal vein is posterior to both of them. That is the arrangement, because anatomy is a subject of order, and you're going to see these structures on ultrasound arranged like that. The hepatic artery is on the left, the bile duct is on the right, and the portal vein is posterior to both of them. That is the arrangement of those structures at the hilum. So here we have peeled away part of the hepatoduodenal ligament to show you deep how those structures are arranged. Now we are going to the next part of the liver, which is also very important. Histology of the liver. When we are looking at histology, we are looking at tissues and cells. When you're looking at the liver histology, or when we look at the word histology, we're looking at tissues and cells. And as I said at the beginning, each part of this lecture is equally important. How is the tissue and the cells of the liver arranged so that they give us a clue of how they perform their functions? How are the tissues of the liver and the cells of the liver arranged so that we can get a clue about their functions? So, the liver is intraperitoneal. It is entirely surrounded by peritoneum with the exception of very few areas. So when you look at that liver, it is lined by visceral peritoneum. When you go to buy liver, some of you who eat liver, who enjoy eating liver, there is a membrane, there is a transparent membrane you can peel off from the outer surface of the liver. I don't know whether you have observed that. There is a transparent membrane you peel off from the outer surface of the liver. That is visceral peritoneum. So the liver is surrounded by that visceral peritoneum. After peeling off that visceral peritoneum, deep to the liver, there is another layer of connective tissue. Deep to the liver, there is another layer of connective tissue, and this is where we need to pay attention. So that connective tissue layer is what we call Glison's capsule, or the fibrous capsule. And we are going to look at the importance of this Glison's capsule, which is very important medically and sonographically and radiologically. So let's say that this is our liver cells. This is liver. And I hope that members are following. What covers the liver? The outer cellular layer, that membrane which you peel off, is the visceral peritoneum. So the outer covering of the liver is the visceral peritoneum. Deep to the visceral peritoneum, we have this connective tissue known as the fibrous capsule or Glison's capsule. So the liver is lined by the outer cellular layer, which is visceral peritoneum, and then a deeper connective tissue, which is known as Glison's capsule or the fibrous capsule. After Glison's capsule, you have a space. Now, this space is not visible under normal circumstances. But if the liver ruptures and it bleeds, blood is going to accumulate in this space between Glison's capsule and the liver, and this is known as the subcapsular space. Sub means below, capsular, the capsule. This is the subcapsular space. Blood can accumulate within the subcapsular space if the liver is damaged. But under normal circumstances, the subcapsular space is collapsed. You cannot see any content there. But we're looking at the liver being lined by visceral peritoneum, which makes the outer cellular layer. Deep to it, you have the Glison's capsule, which is the connective tissue or fibrous capsule. Deep to the fibrous capsule is a collapsed space, which is potential. It can accumulate with blood if the liver is damaged, known as the subcapsular space. And after that, you have our liver parenchyma or liver cells. These are the layers which are lining the liver. And that's very, very, very important. So I've talked about the Glison's capsule. And this anatomist, Francis Glisson, studied it between 1597 and 1677, and it is named after him, Glison's capsule of the liver. And I'm going to talk about the importance of this Glison's capsule. Here I'm just showing where it is located. It is below the outer cellular layer, which is the visceral peritoneum, and below it you have the subcapsular space, which is a potential space, and then you have our liver. So this is the outer cellular layer, this is the Glison's capsule, and this is the subcapsular space down there, and then our liver parenchyma or liver cells. Now, some parts of the liver are not lined by the outer cellular layer. There are parts of the liver which are not covered by the outer cellular layer. They are only covered by Glison's capsule. So that those parts of the liver which are not covered by the outer cellular layer or the visceral peritoneum, those parts of the liver are the ones we call bare areas of the liver. The bare bare area of the liver is that part of the liver which is not lined by the outer serous layer, but the only lining is the Glison's capsule. I'm going to give you an example of those areas known as the bare areas of the liver. So here we are seeing that the peritoneum peels off from the liver to form the coronary ligaments. The same applies here. Peritoneum has peeled off from the liver to form the coronary ligaments. It means that this upper surface is not lined by the outer serous layer of the peritoneum. So this upper surface is only lined by Glison's capsule, that surface there. It's only lined by Glison's capsule. So this is what we call the bare area of the liver. On the superior aspect of the liver, where peritoneum is peeling off to form the coronary ligaments, it leaves this area bare without the peritoneum or outer serous layer, and it's only covered by Glison's capsule, and that's what we call the bare area of the liver. So members, are we following? Members, are you still following with me?
Following.
Yes, sir.
Yes.
Following.
We are dissecting the liver piece by piece, and everything must be important and is important. And now we are in the histology of the liver. So next time they talk about the outer serous layer of this peritoneum surrounding the liver, Glison's capsule, subcapsular space, liver parenchyma, bare area of the liver, you must know what they are talking about. Now, let us look at the importance of Glison's capsule. Let us look at the importance of Glison's capsule. This is a real liver tissue where they got a sample of liver, sectioned it, processed it, and photographed it under a microscope. So this is real liver tissue. So what are we seeing? This is the outer surface of the liver, and I want you to take note. This is the outer surface of the liver, and we are seeing this layer of Glison's capsule forming the outer surface of the layer. Where is the outer serous layer? It has been peeled off. The outer serous layer has been peeled off because it's the first layer which lines the liver. Now we are having Glison's capsule. This is Glison's capsule. And of course, the subcapsular space is there, but for you cannot see it because this is normal liver. It only widens out under normal circumstances. So this is Glison's capsule. But what are we seeing? And I want you to pay maximum attention. Glison's capsule is giving branches which penetrate the liver parenchyma. Whenever I talk about liver parenchyma, I'm talking about liver cells. So Glison's capsule is giving off branches which penetrate the liver parenchyma and go deep into the liver. These septa which penetrate the liver are parts or are branches of Glison's capsule. So Glison's capsule gives off branches known as connective tissue septa, which penetrate the liver and enter deep into it. So the liver is penetrated by these branches of Glison's capsule, which we call connective tissue septa. What is the importance of these branches which penetrate the liver parenchyma? These branches of Glison's capsule which penetrate the liver parenchyma. What is their importance? They act as conduits of blood vessels. Blood vessels pass through these septa as they are moving into the liver. So they are conduits, they are roads through which blood vessels pass. So these septa of Glison's capsule, they give the blood vessels a route where they can pass to enter the substance of the liver. So they are conduits of blood vessels and bile ducts. They pass through the septa of Glison's capsule. Number 2, when these septa penetrate the liver, they divide the liver into subunits known as lobules. The septa of Glison's capsule divide the liver into subunits known as lobules. Two major functions. These septa of Glison's capsule act as conduits of blood vessels and bile ducts. Number 2, they divide the liver into subunits known as lobules. Let us see. So this is our liver parenchyma. And you can see the liver is divided into subunits. These subunits are what we call liver lobules. These are the liver lobules. So we are seeing a septa of Glison's capsule, the different septa of Glison's capsule. These are the different septa of Glison's capsule, penetrating the liver, branching and connecting, branching and connecting. And their branching pattern when they reach the liver parenchyma is going to divide the liver into these units. They are roughly hexagonal. These units are known as the liver lobules, and their boundaries are made up of these septa of Glison's capsule, which penetrate the liver from the surface. They branch and reconnect and divide the liver into these hexagonal, roughly hexagonal units known as liver lobules. And therefore, the liver is divided into lobules by these septa of Glison's capsule, which penetrate from the surface. Meaning that each liver lobule, in its boundaries, we are having Glison's capsule septa. And within this Glison's capsule septa, we have blood vessels and bile ducts moving. So now we have reached the point whereby the liver is divided into subunits known as lobules by the penetrating septa of Glison's capsule, which is very important for our understanding of the function of the liver. So we can see these different lobules of the liver, and their boundaries will have these septa of connective tissue, which are branches of Glison's capsule, which penetrated from the surface. And within each septa, you can see those structures are blood vessels and bile ducts. Those structures, they are blood vessels and bile ducts. Here you can see a branch of the portal vein, a branch of the hepatic artery, and a branch of the bile duct. They move within the septa of Glison's capsule, which divide the liver into lobules. Now, these lobules are not of equal size, as you can see. So they are not of equal size. On rough estimate, they are hexagonal in shape, like this one. What else are we seeing within the lobule? In the middle of the lobule, there is a vein known as the central vein. In the middle of each lobule, there is a vein known as the central vein. Some are visible, some are not visible, depending on how the section was made. But in the center of each lobule, we have a vein known as the central vein. So the liver is divided into lobules, which are demarcated by the septa of Glison's capsule, which penetrate the liver parenchyma. Lobules are not of equal size, but they are roughly hexagonal in shape. And in the center of each lobule, you have a vein known as the central vein. So the lobule is made up of boundaries, made up of Glison's capsule, a central vein, and within it you have liver cells. That is how Glison's capsule is important in the segmentation of the liver into rosy liver lobules. So I say that in the middle of each lobule you have a vein known as a central vein, and we are going to see how important that central vein is. So this is just showing us the blood supply to the liver, that when the structures enter the liver, they divide into right branches and left branches, which acted as our basis for left functional lobe. You can see the left functional lobe is big. And then the right functional lobe, we can see the right functional lobe there. She's showing it to us very well. So the blood which enters the liver through the hepatic artery and portal vein is going to be drained out through what we call hepatic veins. Now these are hepatic veins draining the blood which entered the liver, draining that blood into the inferior vena cava. So blood enters through the hepatic artery and portal vein. So this was showing us that the blood enters the liver through the portal vein and hepatic artery. And then it leaves the liver through hepatic veins. And these hepatic veins drain into the inferior vena cava. So blood has entered the liver through the portal vein and hepatic artery, and it's leaving the liver through hepatic veins, which drain into the inferior vena cava. And you can see the hepatic veins anchoring the liver onto the inferior vena cava and are also acting as supports. When you remove the hepatic artery and the bile ducts, this is our portal vein dividing into the left and right branch. So this is the left functional lobe, and this is the right functional lobe. And then we are having our hepatic veins draining blood out. When you add these two, the image looks like that. And just remember, the artery is on the left, the bile duct is on the right, and posterior to all of them, you have the portal vein. Now, this, before we continue with our histology of the liver, the indication here is that the portal vein is carrying the oxygenated blood, the hepatic artery is carrying oxygenated blood. It means that the liver receives both the oxygenated blood and oxygenated blood. Under normal circumstances, that are your circumstances, the structures of our body must receive oxygenated blood only, and the oxygenated blood is drained out. But for the liver is receiving both the oxygenated blood which enters it through the portal vein and oxygenated blood which enters it through the hepatic artery. So the question would be, why is the liver receiving the oxygenated blood? Because it does not contain oxygen. Okay, it has low levels of oxygen. So why is the liver receiving the oxygenated blood which has low levels of oxygen? This blood is not going to supply the liver with oxygen, but it is going to be filtered. The reason the oxygenated blood is entering the liver is for filtration purposes. It's going to be filtered as it's passing through the liver. It's not going to be supplying oxygen because it's deoxygenated. It's going to be filtered. What is bringing oxygen to the liver cells is the hepatic artery. Because if the hepatic artery are to filter this blood, if the liver is to filter this blood, if the cells of the liver are to filter this blood, they need oxygen. So the oxygen comes in through the hepatic artery. Blood which is going to be detoxified by the liver cells comes in through the portal vein. And the oxygen those cells are going to use to carry out their functions comes in through the hepatic artery. So the portal vein is just taking blood to the liver to be filtered. The hepatic artery is taking oxygen to those cells so that they can carry out these functions. That's why the liver receives both deoxygenated blood in the portal vein and oxygenated blood in the hepatic artery. So let's continue with our liver lobules. Are you seeing the image? Liver lobule is going to represent the others, because the structure of the liver lobule is in principle the same. So we are going to pick out one liver lobule and study it, and it is similar to the rest of the liver lobules. This is also very, 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 very important. Now let us take one liver lobule like this one. I say that it is roughly hexagonal in shape. What forms the boundaries? These connective tissue septa from Gleason's capsule are the ones which form the boundaries of the liver lobule. What do they contain? They contain branches of the portal vein, branches of the hepatic artery, and then tributaries of the bile ducts. And that is where they pass. And these ones interconnect with each other through those Gleason's capsule septa. And we also say that in the middle of the lobule, we have a vein known as the central vein. And inside the lobule, we have these cells, which are the hepatocytes. These are the cells of the liver, which are known as hepatocytes. So this is how the lobule looks like. The septa containing blood vessels, the central vein in the middle of the liver lobule, and we have the liver cells, which are the hepatocytes, which carry out the functions of the liver. These liver cells are the ones which carry out the important functions of the liver. Now, we said that the liver receives deoxygenated blood through the portal vein. And that deoxygenated blood is going to be filtered, not for the purpose of supplying oxygen to the liver. So let us begin with a branch of the portal vein within Gleason's capsule. This is a branch of the portal vein. And I want you to focus where the cursor is. This is a branch of the portal vein. So this branch of the portal vein, these branches of the portal vein, which we are seeing, they give off small branches which enter the liver lobule, which I am pointing at. These small branches are going to pick blood from the branch of the portal vein, and they are going to take that blood and pour it into the central vein. So blood comes from the branch of the portal vein, passes through the liver lobule, through these small blood vessels, and these small blood vessels are going to pour that blood into the central vein. What are the names of these small blood vessels? These small blood vessels are known as sinusoids. They are known as sinusoids. So deoxygenated blood is going to move from the branch of the portal vein. It will pass through the sinusoids, and it will in turn pour into the central vein of the liver. As this blood is passing through the sinusoid, it is passing between hepatocytes. These are hepatocytes. It is passing in between hepatocytes on its way to the central vein. So as it is passing between hepatocytes, there is exchange of materials or substances between the hepatocytes and the blood. So the toxic substances are absorbed into the hepatocytes. The toxic substances are absorbed by the hepatocytes, and that is part of filtration, whereby the toxic substances are absorbed into the hepatocytes, such that by the time blood reaches the central vein, it is less toxic. Then blood moving from the branch of the hepatic artery is going to enter the sinusoid also. But as this blood is passing in between hepatocytes, it is giving oxygen to these cells. It is supplying these cells with oxygen. And by the time it reaches towards the central vein, it is deoxygenated already because it has given out its oxygen. It goes on giving out its oxygen such that at the end of it all, it can mix with blood which came in through the portal vein, because now both types of blood will be deoxygenated. That these small branches of the portal vein and the branch of the hepatic artery, these small branches which enter the lobule are known as sinusoids, and they are capillaries. And I say that this blood, when it is passing through the lobule towards the central vein, it is being detoxified by the liver cells. By the end of it all, it has less toxins. And this blood which is coming from the branch of the hepatic artery is giving these cells oxygen to perform their functions. And by the time that blood reaches the central vein, it is deoxygenated, so it can mix with this. And all that blood pours into the central vein. At the same time, the liver cells are manufacturing bile. When they manufacture bile, they pour it into these small bile ducts. These small bile ducts, which are within the liver lobule, are known as bile canaliculi. Bile canaliculi. These are the ones which are located within the lobule. So when the liver cells manufacture bile, they pour it into the bile canaliculi, and that bile is going to go and drain into this tributary of the bile duct. So that is how the liver lobule performs its functions, detoxifying this blood, receiving oxygen from this blood, manufacturing bile and pouring it into the canaliculi. Let us now magnify this. Let us get a piece of this liver lobule and magnify it and come up here. What are we seeing here? We are seeing the central vein. And I say that you must pay maximum attention. This is the central vein. This is the branch of the portal vein, a branch of the hepatic artery, and a bile duct. So you can see there is a sinusoid here, which is taking this blood towards the central vein. And I say that as this blood is moving through the sinusoid, it is being detoxified by the liver cells. And by the time it reaches the central vein, it is filtered. And the blood within this sinusoid is giving oxygen to the liver cells, so that the liver cells are able to use it to manufacture energy, which is going to allow them to perform their functions. But by the time it reaches these ends, it's also deoxygenated, and it can mix with this other blood and then also pour out into the central vein. And I say the liver cells in turn manufacture bile, and they pour it into these bile canaliculi, and the bile canaliculi are going to drain into the bile ducts. Now, there's another important point, and this you also must note. Inside the sinusoids of the branch of the portal vein, inside the sinusoids of the branch of the portal vein, we have white blood cells known as Kupffer cells. These white blood cells are known as Kupffer cells. They are inside, they are resident inside the sinusoids of the branches of the portal vein. What are these Kupffer cells? The Kupffer cells are white blood cells. And these white blood cells are known as macrophages. They engulf bacteria which are within the blood passing through this sinusoid. And that engulfing of bacteria is part of filtration. So filtration involves removal of toxic substances and removal of bacteria by these macrophages known as Kupffer cells. When these Kupffer cells are still in saturation, they call them monocytes. But when they move to the liver, those monocytes are known as Kupffer cells. They increase in size, they become macrophages, and they are resident in the sinusoids of the portal vein, and they engulf bacteria, which is one of the processes of filtration. So I wanted to bring out that inside these sinusoids from the branch of the portal vein, we have those Kupffer cells, which are engulfing bacteria, which is part of filtration. On top of these hepatocytes removing the toxic substances. And you know the blood from the portal vein is coming from the GIT, the spleen, the pancreas, you remember very well. Now this is another point. Between the liver cells, the hepatocytes, and the sinusoids, there is a small space there. There is a small space there. This small space is between the sinusoids and the hepatocytes. And I say that when substances are moving from the sinusoids to the hepatocytes, they traverse that space. When substances are moving from the sinusoids to the hepatocytes, they traverse that small space. This small space between the sinusoids and the hepatocytes. That small space is known as the space of Dise. Space of Dise. Or you can call it perisinusoidal space. This space is known as the sinusoidal space or space of Dise. That is the space between the sinusoids and the hepatocytes. And that is where exchange of substances between the hepatocytes and sinusoids takes place. The substances cross the space of Dise as the blood is being detoxified. That space of Dise is also very important. And we are going to look at it in detail. So this is a simplified illustration showing us the central vein, the branch of the portal vein, the branch of the hepatic artery. You can see the sinusoids taking that blood towards the central vein. This blood is oxygenating, this blood is being detoxified. The cells of the liver manufacturing bile and draining through the bile current right to the bile duct. And you see at the end of it all, blood in the which came in oxygenated is going to mix with this blood which is deoxygenated, and all that blood is going to pour out into the central vein. And then you're seeing those Kupffer cells which are resident in the sinusoids to filter the blood by removing infections or bacterial infections. So when blood enters the central vein, it means that all lobules have central veins. So when blood enters the central vein, which we have seen there, when blood has entered the central vein, from the central vein, that blood is taken to what we call the interlobular veins. Blood from the central veins, because remember we have very many central veins because we have very many lobules. This blood is going to enter into what we call the interlobular veins. Then the interlobular veins are going to connect to form the hepatic veins, which hepatic veins will drain into the inferior vena cava. So blood from the central vein drains into, blood from the central veins is going to drain into what we call interlobular veins, and the interlobular veins are going to drain into hepatic veins. We have 3 hepatic veins. So the interlobular veins are going to confluence to form 3 hepatic veins, and those 3 hepatic veins are the ones which are going to finally drain into the inferior vena cava. So the perisinusoidal space is known as the space of Dise. The perisinusoidal space is also known as the space of Dize, which is the space that I talked about. That is the space of Dize, or peri, on the sides of the sinusoids, perisinusoidal space or space of Dize, where we have substances crossing between the blood in the sinusoid and the hepatocytes, and also on this side, oxygen crossing. Now, this space of Dize, apart from acting as a medium where we have exchange substances are crossing, this space of Dize also contains some cells. The space of Dize contains cells known as the hepatic stellate cells, or you can call them perisinusoidal cells because they are found in the perisinusoidal space, or Ito cells. These cells are located in the space of Dize. So on top of the space of Dize, acting as a medium through which substances cross between hepatocytes and sinusoids, it also contains cells known as hepatic stellate cells, or perisinusoidal cells, or Ito cells. What is the function of these stellate cells, which are located in the perisinusoidal space? They are involved in liver fibrosis. What is liver fibrosis? Liver fibrosis is a process by which damaged liver cells are converted into scar tissue. When your body is damaged, scar tissue is formed. So when your liver cells are damaged, they are converted into scar tissue. This usually occurs where we have livers of the alcoholics, chronic alcoholics. Chronic alcoholics have a disease which is known as liver cirrhosis. Chronic alcoholics get a disease known as liver cirrhosis. Liver cirrhosis is where part of your liver becomes fibrous or becomes scar tissue. It is because when the liver cells are damaged, they die off and they are converted into fibrous tissue, which is the process known as fibrosis. For example, an example of liver fibrosis is a disease known as liver cirrhosis, where part of the liver becomes scar tissue. So the hepatic stellate cells, which are located in the space of Dize, are involved in the conversion of damaged liver cells into scar tissue, process known as liver fibrosis. And fibrosis is a process of healing, because when a scar is formed, that is healing. But the danger in that is that you have lost liver cells, therefore you have diminished the function of your liver, and this scar tissue can block the blood vessels, especially the branches of the portal vein. This scar tissue can block the sinusoids and prevent blood from passing through the liver normally. Then blood is going to accumulate in the portal vein and cause what you call portal hypertension. And then it's going to find alternative ways like the anterior abdominal wall causing caput medusa, the lower third of the esophagus causing esophageal varices. So whenever the liver cells are damaged, they are converted into fibrous tissue, and these cells known as hepatic stellate cells, which are located in the space of Dize, are important in the conversion of damaged liver cells into scar tissue. Scar tissue is a process of healing, but it has consequences. And I said chronic alcoholics, because the liver overworks to detoxify the blood of alcohol. The liver overworks. And when the liver overworks, it enlarges, it hypertrophies. And then when it continues to overwork, the liver cells break down and they are converted into scar tissue by the hepatic stellate cells, which are located in the space of Dize. We are breaking down the liver piece by piece in detail. Similar example, central vein, bile duct, portal vein, hepatic artery. You can see the sinusoid. Blood is going towards the central vein, pouring there. And then inside you can see the Kupffer cells. Then this is the space of Dize. And in the space of Dize, you're seeing these hepatic stellate cells are there. Those are the hepatic stellate cells, these ones within the space of Dize. And you can see the canaliculi bringing bile out of the liver into the bile duct. This is a magnified illustration of that part. So we have finished the histology of the liver. We have finished the histology of the liver.